Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
I want you to start by visualizing something for me. Picture your brain, but not as this static fleshy organ that you see in a jar in science class.
Right.
I want you to think of it as a piece of incredibly advanced adaptive software. It's dynamic. It's responsive. It's constantly patching itself, updating its own drivers, and rewriting its own code based on everything you give it every single day.
It's the ultimate learning machine, really. It adapts to whatever environment you place it in, for better or for worse.
For better or for worse, that's the key. So, okay, now imagine a specific script that's running on that software, and it's running in the background, just quietly, persistently, day in and day out. Let's say this script runs for 35 years. And that script is the chronic consumption of alcohol. And just to be really clear here, we are not talking about, college weekends or the occasional glass of wine with dinner.
No, this is a steady, decades-long input. And when you run a script like that for 35 years, you're no longer just dealing with a habit, you're dealing with a fundamental architectural change. The software, it eventually alters the hardware itself.
And that is the terrifying, but also kind of fascinating premise of the deep dives we are getting into today. We're looking at a landmark study that was just released this week, February 2026. It was published in the journal Addiction.
The title is Decades of Drinking Reshape Gene Expression in Key Human Brain Regions.
And I have to say, when I first saw that title, I thought, okay, here we go. Another study telling us drinking is bad for us. We know that. But this is different.
It is significantly different. This research comes out of the Institute for Neurosciences in Spain. The lead researchers are Dr. Maria Salud Garcia Gutierrez and Dr. Jorge Manzanares. And what they've done here is they basically provided a molecular map of exactly which switches get flipped in the human brain after a lifetime of alcohol use.
So today, we're going to find out why the term willpower might be, well, a biological oversimplification when we're talking about long-term addiction.
A massive one.
We're going to look at how the brain actually builds more roads leading to craving, while at the same time, it's tearing down its own defense walls.
It's kind of biological double cross. The brain literally turns against its own well-being, all in a desperate attempt to maintain what it now thinks is equilibrium.
Before we get into the mechanics of that, which are pretty scary, we need to talk about how they even figured this out. Because in science, and I think especially in addiction science, the who you study is just as important as the what you find.
Absolutely.
And this study claims to have a gold standard sample. What does that even mean? Why is this sample so special?
Well, to really get why it's so special, you first have to understand how incredibly difficult it is to study the brain of someone with what we call alcohol use disorder, or AUD.
Because in the real world, addiction is messy.
Exactly. It's rarely a solo act. You might have someone with an alcohol problem, but they also have a pack-a-day cigarette habit.
Or maybe there's cocaine use on the weekends, or maybe opioids are involved. It's a mix.
Precisely. And from a research perspective, that's a complete nightmare. If you look at a brain and you see that a certain gene is, let's say, broken, how do you know what did it? Was it the whiskey? The nicotine? The painkillers? You can't know.
It just muddies the data. You can't isolate the variable you're trying to study.
You can't. So what you need is a pure sample. You need to find people who drank heavily for decades, but somehow never touched anything else.
Which sounds like, that's finding a needle in a haystack, right?
It's nearly impossible in most parts of the world, but this research team, they utilized the New South Wales Tissue Resource Centre in Australia. And this is the crucial part. Australia, it seems, has a specific demographic pocket where you can actually find individuals with severe chronic alcohol use disorder who do not have a history of abusing other illicit drugs.
So they found the unicorns of addiction science.
They essentially did, yes. So they compared post-mortem brain tissue from 18 men with this pure AUD against 18 healthy controls. And the statistic that really, it just stops you in your tracks, is the duration.
Yeah.
The average history of alcohol consumption in the AUD group was 35 and a half years.
35 and a half years. That's an entire adult life for some people.
It is. And that creates this unique scientific opportunity. Because we've eliminated the other variables, the other drugs, and because the duration is so incredibly long, we can say with very high confidence that the changes they're seeing in the brain, these are the specific genetic footprints of alcohol. This is what ethanol and ethanol alone does to the human brain over 3.5 decades.
Okay, so we have our pure sample, the who.
Yeah.
Now we need to define the target, the what. The researchers didn't just, put the whole brain in a blender and look at the soup. They zoomed in on one very specific system.
The endocannabinoid system.
Now, I know when people hear that word, they immediately think of cannabis.
Which is completely fair, because that's actually how we discovered the system in the first place. But the name is a bit of a misnomer. The endocannabinoid system is ancient. It's been in our biology long, long before humans ever discovered the cannabis plant.
So what does it do?
Think of it as the brain's fine-tuning system.
I like that analogy, the fine-tuner.
Yeah. If your brain is a massive orchestra, you've got the percussion of your motor skills, the strings of your emotions, the brass section of your executive planning.
Okay.
The endocannabinoid system is the conductor who walks around and makes sure everyone is playing at the right volume. It regulates mood, it regulates stress, it regulates memory, and, most importantly for our discussion today, it regulates reward and motivation.
So to understand the damage that alcohol does, we have to understand the tools this conductor uses. The study breaks it down into 3 categories, which I think is a great way to simplify it because the names can get pretty technical.
Yeah.
So let's think of them as the locks, the keys, and the janitors.
That is probably the best way to visualize it. Let's start with the locks. These are the receptors. They sit on the surface of your brain cells just waiting for a signal. In this study, we're looking at 2 main locks, CB1 and CB2.
Okay, CB1 and CB2, those are the locks. So what are the keys?
The keys are what we call ligands. These are the chemicals your own body makes naturally, endocannabinoids, that float across the gap between cells and fit perfectly into those locks. When the key turns the lock, a message is sent. A message like, calm down, or feel good, or forget that stressful thing that just happened.
And then finally, you've got the janitors.
The janitors are the enzymes. In this study, we're mostly looking at 2 called FAAH and MGLL. Now, once the key has turned the lock and the message is sent, you don't want that signal firing forever. You need to reset the system. So the janitor comes in, sweeps up the chemical key, and breaks it down, readying the lock for the next signal.
So in a healthy brain, this is a perfect self-regulating cycle. Key turns lock, message is sent, janitor comes and cleans up. The system is ready for the next message.
Exactly. It's a beautiful homeostatic balance. It keeps you level.
But the researchers, they wanted to see what happens to this delicate balance in 2 very specific rooms of the brain. And these aren't just random spots. You could argue they're the 2 most important rooms when it comes to our behavior.
They looked at the prefrontal cortex and the nucleus accumbens.
Let's define those before we dive into the damage. The prefrontal cortex.
That's the CEO of the brain. It sits right behind your forehead. This is the center of your executive control. It's responsible for judgment, for planning, and critically, for inhibition. It's the part of the brain that says, I really want that second piece of cake, but I'm trying to be healthy, so I will not eat it.
It's the brake pedal.
It is the biological brake pedal.
And the nucleus accumbens.
That is the reward hub. It's deep in the center of the brain, a much more ancient primal part. It's responsible for motivation, for pleasure, for habit formation. When you do something that ensures your survival, like eating or finding a mate, the nucleus accumbens lights up to say, hey, that was good. Do that again.
So it's the gas pedal.
It's the gas pedal.
Okay, so we have the CEO, the brake pedal, and we have the reward hub, the gas pedal. And we are looking at the brains of men who have been flooring the gas with alcohol for 35 years. The central question is, what happened to the locks, the keys, and the janitors in these 2 specific rooms?
The results paint a picture of a system in total chaos. The balance is completely and utterly gone.
Let's go through the 4 major findings, because each one seems to peel back another layer of this biological tragedy. Finding number 1 involves the CB1 receptor, the first lock we mentioned.
Right. The CB1 receptor is the most abundant of these receptors in the entire brain. And we know from a lot of other research that when you stimulate CB1, it powerfully reinforces behavior. It drives that feeling of wanting. It is a core part of the reward-seeking pathway.
So what did the study find in the brains of the 35-year drinkers?
Well, in the control group, the healthy brains, the levels of CB1 were normal. But in the alcohol group, the gene expression for CB1, which is basically the instruction manual to build this specific lock, had skyrocketed. In the prefrontal cortex, the CEO's office, it was increased by 125%.
125%. That's more than double.
It's more than double. And in the nucleus accumbens, the reward hub, it was up by 78%.
I want to really sit with that for a second. We're talking about gene expression here. So this means the cells themselves are reading the DNA code and frantically printing out more and more and more of these wanting receptors. What's the functional result of that? What does it feel like to have double the amount of these locks?
Think of it like volume control. If a healthy brain has, say, 10 speakers playing the message, "I would like a drink," the brain with chronic alcohol use disorder has physically built 20 more speakers and turned them all up to maximum volume. The brain has restructured itself to be hypersensitive to the reward of alcohol.
So when we talk about someone having a relapse or struggling to quit, it's not just a thought that they're having. It's not a simple choice.
No. It's a biological scream. The lead author, Professor Garcia Gutierrez, she specifically links this incredibly high level of CB1 to the vulnerability of relapse. The brain has literally built an infrastructure that mans the substance. The little country road to addiction has been widened into a 10-lane superhighway.
That is terrifying enough on its own. But then we get to finding number 2, which I have to say I think is even more disturbing, and this involves the other lock, CB2.
Right. If CB1 is the craving switch, CB2 is the defense shield.
The defense shield.
Yes.
Wow.
Unlike CB1, which is all about signaling and reward, CB2 is largely responsible for neuroprotection. It regulates inflammation. When your brain is under attack, and let's be clear, alcohol is a neurotoxin, so it is an attack, CB2 is part of the immune response that tries to keep the tissue healthy and manage the damage.
It's the fire department.
It's the fire department. It rushes in to manage the inflammation so the cells don't die.
Okay, so in the brains of these men who have been essentially setting a fire every day for 35 years, you would hope, or at least you would logically expect, that the fire department has been beefed up, right? That the brain would build more defenses.
That is what logic would dictate. But the study found the absolute opposite. In both the prefrontal cortex and the nucleus accumbens, the gene expression for the CB2 receptor, it dropped by approximately 50%.
It dropped by half.
By half.
That feels like a complete paradox. Why on earth would the brain delete its own defense system while it's actively being poisoned?
And that is the absolute tragedy of what we call maladaptive plasticity. The system is either just exhausted or the alcohol signaling has so profoundly disrupted normal function that the brain stops producing the very thing it needs to survive. So now just look at the combination of these two findings.
Okay.
You have the craving lock, CB1, turned up by 125%. At the same time, you have the defense lock, CB2, turned down by 50%. It's a perfect storm. The walls are crumbling, the fire department has been sent home, and the person inside is biologically screaming for more gasoline.
That is a very apt, if grim, metaphor. And it helps explain why we see such profound cognitive decline and physical damage in long-term alcoholism. It's not just the alcohol directly killing the cells, it's that the brain has lost its own ability to manage the inflammation caused by the alcohol. The shield is broken.
Now, there was a third receptor they looked at, one they called the Orphan, GPR55. Why do they call it an orphan?
In science, we call a receptor an orphan when we've identified the lock, but we don't know who its parents are, meaning we don't yet know which natural chemical key in the body is supposed to open it. We're still in the process of figuring out what GPR55 actually does.
And this study is apparently the first time anyone has documented changes in this orphan receptor in humans with alcohol use disorder. And the behavior of this orphan was... well, it was weird.
It was very strange. It was what we call region-specific. So in the prefrontal cortex, the CEO's office, the orphan receptor was up 19%. Okay, a modest increase.
But in the nucleus accumbens, the reward hub, it crashed. It went down 51%.
So why would it go up in one room and down so dramatically in the other?
That is the million-dollar question. We have some clues. We know GPR55 seems to be involved in inflammation, maybe in pain regulation. The fact that it acts so differently in the executive center versus the reward center suggests it plays a really complex, nuanced role. It could be that in the CEO office, it's trying to compensate for the crazy high CB1 levels, trying to restore some order. But in the reward hub, it's just collapsing under the pressure, just like the CB2 shield.
So it just adds another layer to this picture of a system that is completely confused. It's misfiring all over the place.
It creates a confused and contradictory signaling structure. The brain doesn't know whether to protect itself or to lean into the craving.
So we've talked about the locked CB1, CB2, and the Orphan. Now we have to talk about the Janitors, the enzymes. This was finding number 4.
Right. We're looking at FAH and MGLL. And remember, their job is to clean up the feel-good chemicals, especially one called anandamide, which is sometimes called the bliss molecule. If the janitor works too slow, that bliss signal lasts a long time. If the janitor works too fast, the pleasure is cut short.
So what are the janitors doing in the alcoholic brain?
Well, the study found that FAAH, the main janitor for that bliss molecule anandamide, was significantly higher. Specifically in the nucleus accumbens, it was up 24%.
Okay, so in the reward hub of all places, the janitor is working overtime. He's like manically scrubbing the flowers.
Exactly. He is scrubbing the floor before the party's even had a chance to start.
And what's the consequence of that, functionally?
It means that any small natural pleasure, the little bit of anandamide your brain produces when you see a friend or you eat a good meal or you see a beautiful sunset, it gets instantly wiped away. It is degraded and removed before it can even register as pleasure.
That's just heartbreaking. So the natural high of just being alive becomes chemically impossible to sustain.
It creates a state that researchers call a reward deficit. And if your baseline for a natural pleasure is basically 0 because the janitor is too aggressive, what do?
You look for something that can overwhelm the system.
You look for something that creates a massive artificial flood of reward that can temporarily overwhelm that overactive janitor. You reach for the bottle.
It really connects all the dots. The enzyme imbalance actually drives the seeking behavior.
Yeah.
You're not even drinking to feel good anymore. You're drinking just to feel normal because your natural system is being scrubbed clean before it can work.
It completely changes the paradigm from seeking pleasure to escaping the deficit.
So just to recap the mechanics here, because it is a lot to take in. After 35 years of chronic alcohol use, the brain has 1, more than doubled the craving receptors.
TB1.
2, it's halved its own defense receptor.
TB2.
3, it's got this confused orphan signaling system, GPR55, that's going in opposite directions. And 4, it has put the janitor on hyperspeed in the reward center, which kills off any chance of feeling natural pleasure.
It is a machine that has been completely and meticulously re-engineered to do one thing: sustain addiction and suppress recovery.
Which brings us to the implications. Why does this matter? I mean, obviously it matters to scientists, but why does it matter to the person listening right now, or maybe the family member of someone who's struggling?
It matters because it moves the entire conversation away from morality and places it squarely in the realm of biology. For decades, for centuries really, society has viewed alcoholism as a failure of character. Why don't they just stop? Why don't they have more self-control?
Right, the whole just say no idea.
But when you look at these results, specifically that 125% increase in CB1 receptors in the prefrontal cortex, you begin to realize that saying no might be functionally impossible for this brain structure. The executive control center, the very part of the brain you need to make that decision, has been physically rewired to scream yes.
It's not that they won't use the brakes on the car. It's that the brakes have been taken out and replaced with more gas pedals.
That's a perfect way to put it. And this validates what Professor Jorge Manzanares, one of the study's authors, said. He noted that our current treatment options are pretty limited. We've been using this one-size-fits-all approach, often just trying to sedate the patient or use drugs that make them sick if they drink.
But this data suggests we can be much, much more precise.
This is really the dawn of precision medicine for addiction. If we know that CB1 is too high and CB2 is too low, we don't just need a generic brain pill. We need a therapy that can specifically block CB1 to lower that craving volume while simultaneously stimulating CB2 to help rebuild the brain's shield.
You could, theoretically, tune the knobs back to their original factory settings.
That is the ultimate goal. And the fact that we see these changes are specific to certain regions, that the PFC is different from the AC, means we might eventually develop drugs that can target those specific areas of the brain. We are not there yet, not even close, but this research gives us a map. It shows us where to go.
I do want to play devil's advocate for a moment, though. We are looking at a study of 18 people. That's a small number. Is that really enough to draw these huge conclusions?
No, you're right to be skeptical, and that's good science. In the world of big data, 18 seems like a tiny number. It is a limitation of the study, and we have to be careful about generalizing these exact percentages to every single person on the planet.
But the argument is that the quality of the sample balances out the small quantity.
In this specific case, yes. Finding 18 pure cases with 35 years of history is, from a data perspective, statistically more significant than finding 1,000 messy cases with mixed drug use. The signal is incredibly clear, even if the group is small. But does it need to be replicated? Absolutely.
And it needs to be replicated in women.
100%. This study was exclusively on Caucasian males. We know for a fact that female brains often metabolize alcohol differently. We know the endocannabinoid system interacts with hormones like estrogen. It's a massive blind spot in the research. We cannot just assume that a female brain would show the exact same 125% increase. It might be higher. It might be lower. It might involve different receptors entirely.
And then there's the classic chicken or the egg problem. Since these brains were all studied post-mortem, after death, we're looking at the end of the movie. We know how it ends.
Correct. We are seeing the wreckage. The study strongly implies that the 35 years of drinking caused these genetic changes. The correlation is powerful. However, we can't get in a time machine and scan these men's brains before they took their first drink.
So is it possible that maybe they had high CB1 levels to begin with? That they were just genetically prone to addiction from the start?
It is possible, and in all likelihood, it's probably a mix of both. They might have started with a slight genetic predisposition, a slight imbalance, and then the 35 years of alcohol exacerbated it into a chasm. But given the sheer magnitude of these changes, that 125%, it is highly unlikely that was all there at birth. The alcohol was the primary driver of the remodeling.
There was one other confounding factor mentioned, which was depression.
Yes, and that's an important one. Many of the subjects in the AUD group also had a clinical history of depression. And we know that depression on its own can also mess with the endocannabinoid system. So there is a layer of complexity there. Is that low CB2 shield a result of the alcohol or the depression or the interaction of both? It's very hard to untangle.
Even with those limitations, though, the overall picture is remarkably consistent. It's a picture of a brain that is adapted to a toxic environment by, in a way, becoming toxic to itself.
It really highlights the double-edged sword of what we call neuroplasticity. We love to talk about plasticity when we're, learning a new language or recovering from a stroke. We say, look how amazing the brain is. It can rewire itself.
But this is the dark side of that same coin.
This is maladaptive plasticity. The brain is agnostic. It doesn't have a moral compass. It doesn't care if the input is learning French or drinking a liter of vodka every day. It will rewire itself to become more efficient at processing that input. In this case, it optimized itself for addiction.
It optimized itself for addiction.
Yeah.
As a haunting phrase, it implies the brain is actually working perfectly. It's just working toward the wrong goal.
That is exactly what is happening from a biological standpoint.
So as we wrap up this deep dive, I want to circle back to that number that's been sticking with me this whole time, 35 years.
It's a powerful reminder that biology is a very long game. We tend to think of our choices in the moment. I'll have a drink tonight. I'll start that diet tomorrow. But gene expression is a cumulative project. The script that you run today, if you run it enough times, becomes the software of tomorrow.
And eventually becomes the hardware.
And eventually it becomes the hardware.
But the flip side of that, and I think we have to end on a glimmer of hope here, is that if the brain is plastic enough to wire itself into this state,
then theoretically, with the right inputs, enough time, and maybe with the help of the future precision medicines this study points toward, it can be rewired out of it. The system isn't permanently broken, it's just set to the wrong frequency. And knowledge, like what we've discussed today, is the very first step to figuring out how to retune it.
for everyone listening, whether you're just geeking out on neuroscience with us or this topic hits a little closer to home, I hope you take this as a call to view addiction through a lens of mechanics, not just morality. These are gears and levers. And we're finally starting to figure out how they work.
And that is the most hopeful thing science can possibly offer.
Thank you for going on this journey with us. It's heavy stuff, but it's important. Stay curious, take care of your hardware, and we will see you on the next deep dive.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.